Okay, we're officially recording. Again, for those of you who just joined, welcome to day 8 of the boot camp. So just 2 days left. Thank you guys so much for hanging out with us. I know it's been a lot of material. I just wanted to go over the schedule for today and tomorrow, because I did finally put in a schedule for tomorrow now that we know where we're at with the content. And so we'll start, as we kind of already have, with just questions from yesterday or any of the material that we went over so far. And then in about 10 minutes, we'll have Armin get started to give a presentation on the work he's been doing at Planetary Protection in the realm of metagenomics. We'll then have our break a little bit earlier today. So our break will be at 9:10 to 9:30. And then when we get back from break, we'll hear from Lisa and some of the work that she's been doing in Planetary Protection and some details about processing metagenomics data. We'll then get right into a debrief on that. So we'd love to hear feedback from you guys about Armin and Lisa's presentations. If there's anything from their presentations that you would like to see added to or that you would like to take to implement in your own home institutions. And then we'll go into lunch. And then after lunch, we're going to pick up where we left off yesterday with the DGE Jupyter notebook. And then once we're done with that, we're going to start our debrief on both the stats lecture that Lauren gave and the DGE Jupyter notebook that you guys are going through with Lauren. And then we'll have our break, and then we can carry on with the debrief if we haven't finished. But then we want to give you guys time to revisit the intro Jupyter notebooks, the Unix and R intro Jupyter notebooks. And I know some of you guys talked about adding some things that would be useful for your students that you would like to see. And so we want to give you guys time to do that. So we'll revisit those Unix and R Jupyter notebooks, give you guys some time to go back through those again, add things that you think are going to be useful for your students that we might not have put in there. And then you'll be able to save those notebooks, and we'll set up a location for you to save a final copy of them. So that way your modified versions will be the ones that you'll be able to use when you teach at your home institutions. And then we'll end for the day. And then tomorrow, our last day of the boot camp, we'll first, as we do every day, start with questions. And then we'll revisit the FastQ to Counts Jupyter notebook and give you guys the opportunity to make any changes, additions, modifications to that notebook. And of course, we'll all be here. So if you guys have questions, need help making anything, or want us to help add any content that you think would be useful that isn't already there, go ahead and do that, and then we'll get into our break. We'll have our break at our regular schedule for the morning tomorrow at 9:30. And then we'll come back from break and give you guys time to modify the DGE Jupyter notebook in any way that you guys see fit. And then we'll go over how to sign up for resources for SMCE to make sure that that's super clear for everyone, and how to set up, once you have all of your students' accounts there and accessible, how to get started with the Jupyter notebook and the hands-on materials. So we'll go over all that before lunch tomorrow. And then after lunch, we'll just make sure that everyone is super clear on how they're planning to implement the bootcamp material at their home institution. Make sure that you guys have everything you need from us to be able to do that. And then I'm going to give you guys time to take the post-bootcamp survey. And so there's a post-bootcamp survey, so we'll all do that together during the bootcamp. And then after that, we're going to do some of the extra stuff that you guys had talked about wanting to do. So Lauren and Mike graciously said that they would go over how to install Conda, Jupyter, and R locally. So we'll give you guys all the opportunity to install those on your local computer if you would like. And then we'll have our regular scheduled afternoon break. And then we'll come back and I'll show you that once you have those installed locally, how you would say download the genes.results files from RSEM from any data set you want to on GeneLab, and be able to run our DGE script locally through our studio. And then we'll have a wrap up with any final questions and comments. Any questions about the schedule? Is there anything that you guys wanted us to go over that isn't on the schedule? Okay, great. So with that, was I sharing my screen? I wasn't sharing my screen at all that whole time, was I? Whoops. Oh, yeah. That's what you guys should have been looking at with me. Sorry. But all this stuff can be found here. So this is basically everything that I just went over. for the schedule. Okay, so yeah, I put the link in the chat to the schedule, just went through everything verbally. Sorry, I didn't have that up. So I'm gonna go ahead and stop sharing now. And then just, are there any other questions about anything that we've gone over so far in the boot camp? You can unmute your mic, type it in the chat. Okay, we'll have more time for the answer questions stuff during the debrief section as well. So with that, I'm going to hand it over to Armin to go ahead and present about his work in JPL's planetary protection. Take it away, Armin.
Thank you, Amanda. Share my screen. All right, hello everybody. We're going to switch gears pretty dramatically from, can everyone see my screen?
Looks good.
Okay. You're showing the
presenter view?
Yeah.
Let's do this. How about now?
That's good.
Okay. All right, so we're going to be switching gears pretty dramatically from RNA sequencing. I'm going to be talking about two different projects that we've been working on for the past couple of years at JPL. This is actually with Lisa as well. Feel free to stop me at any time and ask questions about any of the slides. I'm going to assume that you all remember a little bit from the first slide that Lisa presented on sort of the introduction to planetary protection and some of the background. But if there are any questions pertaining to that, again, feel free to stop me. So the first project is about far UVC applications to spacecraft microbial reduction. So sort of going through the current methods that are being used, the main primary technique that we use is dry heat for doing microbial reduction on spacecraft hardware. And the issue is that that technique does not mitigate ongoing contamination that's happening throughout the assembly activities and launch. It's something that needs to happen sort of after the spacecraft's assembled. So this diagram is just showing if we start sort of with hypothetically dirty hardware, planetary protection comes in and we do a bioassay to establish the sort of base microbial bioburden that's there. And then there's some sort of reduction and precision cleaning that happens, and we arrive at a cleaner piece of hardware. And then once that individual piece of hardware is cleaned, it's then taken into some sort of assembly operations and integrated with other pieces of hardware. And as you can imagine, that process can introduce contaminants because there's engineers sort of handling the pieces. And so there's sometimes a need for another bioassay at that point to establish if we veered away from the original sort of cleanliness. And then if that bioassay also passes, then again, it moves along in the assembly process and ends up on the launch site, which again, there's some more sort of handling and manipulation that happens there. Anytime there are humans around, there's a risk for recontamination. And as we get sort of closer and closer to launch, the opportunities to perform bioassays become more and more difficult. And so there's this sort of unknown or sort of modeled bioburden that happens in between sort of the potential launch recontamination and sort of how clean the final hardware is as it arrives to the destination. So that was sort of the motivation that drove the project. Sort of the objectives was to look at this new technology. UVC is obviously not new. It's been used for many, many years, and we know the sort of capabilities in microbial reduction. But a team out of Columbia recently started using a new wavelength. So the traditional wavelength that's used is 254 nanometers, but they started using 222 to inactivate microbes. And the real advantage to this wavelength is it's not harmful to humans. whereas 254 causes skin damage and obviously eye damage. And so it's really exciting because it provides an opportunity to use this technique in the presence of humans. The motivation for doing the work at JPL was to test some of our hardy spore-forming microorganisms and to see if this technique is sort of efficacious on those organisms, since Columbia had established that it works with some of the vegetative cells and more sort of medically relevant species. So we wanted to test on Bacillus pumilus, SAFR 32 from the name. I don't know if that rings a bell to anybody, but SAF is the spacecraft assembly facility. It's one of the clean rooms that we have at JPL, and this organism was found there by one of our group members years back and identified. And since then, it's the absolute champion of resistance for UV. So the organism's been sent up to the ISS and exposed, and that's what I'm showing on the right. This is a study that you all can look up. So they basically sent spores of this organism to the International Space Station. It was spiked on coupons. And it was exposed outside the space station under various different conditions. One was completely open to sort of space UV and vacuum and all the other environmental conditions. Others were sort of filtered to mimic the UV that you'd be seeing on the surface of Mars. And the exposure lasted for 18 months. And then the coupons were sent back down. I don't show the survivability, but there were a surprising amount of spores in most of the conditions. I think the space UV was the only condition where all of them died. But under the Mars condition and some of the dark UV conditions, there was some pretty high survivability. The other organism we want to work with was Acinetobacter radioresistens, another really well-known sort of UV and resistant bug that's also resistant to hydrogen peroxide. I'm showing a chart on the bottom right of that organism from a different study where they exposed it to both desiccation, UV, as well as hydrogen peroxide. And again, you can see the sort of survivability of this organism. And this is not a spore-forming vegetative cell or organism, and so it's actually the vegetative cells that are surviving these conditions, which is even more impressive. So we want to characterize the effects of varying dose rate on the efficacy of the technique, and then ultimately the goal was to establish this as a surface microbial reduction tool for PP. So this is some of the preliminary results from our study. We did the pilot experiment 2 years back, and we were able to show that this technique works for both Bacillus pumilus spores as well as vegetative cells of Acinetobacter. You can see there's a sort of steep reduction that's happening in the beginning, up to 50 millijoules. And then beyond that, this sort of tailing effect is typical of most reduction techniques. It's not too well characterized as to why. Sort of some of the theories are that there's shielding happening on the coupons. So some of the dead organisms on top are shielding living ones from the bottom, or they're due to sort of strain or individual cell level variations in resistance. Nonetheless, this is sort of a promising initial sort of reduction that we're seeing. The methods that we used, so we wanted to mimic spacecraft hardware and sort of real life conditions as much as possible. So we went ahead and used aluminum coupons, which are commonly used as a proxy for spacecraft hardware. We spiked about 10 to the 5 CFUs total on each coupon prior to exposure. And then we recovered them using a PVA technique, sort of peeling the organisms off and resuspending them in solution and plating them out. And then on the right, I'm just showing the power meter that was used to make sure that the dose rates and the total dosage was what we intended. This is some data from Columbia, actually prior to us starting this work, where they're showing sort of the top left, they're showing the technique that they had, the filter that they use on the UV lamp is actually, in fact, selecting for 222 nanometers only, and you're not getting any other wavelength. And then on the right is a study that they did with some corneal tissue and exposing it to, again, 222 nanometer far UV. And as you can see, you have pyrimidine dimers forming in 254 nanometers, but you're not seeing that in 222. And so the dimers are obviously a sign of damage or DNA damage. And then on the bottom is a study that they published back in 2020 showing that this technique is also efficacious in inactivating coronaviruses or airborne viruses. So the effects of varying dose rate, we also wanted to sort of characterize and understand that. Again, the starting population that we used was around 10 to the 5 or 10 to the 6 spores per coupon. And that is roughly the equivalent to the sort of bioburden threshold that were allowed on March 2020. We could show that this exposure resulted in 3 logs of kill. And the effects of these 2 different dose rates were not significant, but the chances of sort of having larger dose rate difference resulting in a more significant difference in reduction is highly likely, but we only tested these 2 dose rates. And then for Acinetobacter, like I said, Acinetobacter is a non-spore forming organism. The rate at which we tested this organism was at 0.5. So compared to Psilus pumilus, we actually used 3 different dose rates. And so when we compared the 0.1 milliwatts to the 0.5, there was a significant difference, but obviously that difference we can't attribute exclusively to dose rate since there's an organism difference as well. But we were able to show that. So some of the advantages of this technique requires minimal intervention by engineers. The idea would be to have these UV lamps in the clean room environment as assembly is going on. And so you're continuously knocking down the contamination. It can be implemented within the assembly environment. It can also be used as a recontamination prevention strategy on the launch pad, in the payload fairing during launch, doing things like environmental testing or transport of the spacecraft. So there's tons of applications. And then this is some of the work that is sort of ongoing now. We have another grant to further develop this technique. And so there's some ongoing testing. We want to link the industry standard 254 nanometer reductions to the 222. We want to understand the sort of compatibility across different materials, test a wider range of model organisms, and then establish some protocols for implementing this technique. So the value added, going back to the original chart, the idea is we'll have far UVC everywhere throughout the entire process. But really, I mean, the applications can be tailored to sort of the need. It can be to knock down the microbial contamination from dirty hardware. It can be used to keep clean hardware clean. And then it can obviously mitigate some launch recontamination as well. This is one of the publications that we got out of this work, and hopefully more soon. So that's the end of that first project. I can pause now for any questions, and then I can hop into the next one, if anybody has anything.
Hi, Armand. It's Elba.
Ah, you got cut off, Elba.
Sorry, I'm muted. This is Elba. So I don't know how much you heard, if any. Um, are you at all using nanopore sequencing, or is that a technology that would be useful for this microbiology mission that you're on?
Yeah, so we actually we got a MinION a couple years back when it first came out. It was very exciting, and I was actually handed the instrument from my PI saying, "See what you can get out of this." And we did use it. So we don't have sequencing capabilities in-house. Almost, I mean, pretty much all the sequencing that we do is through vendors. Um, but there were a couple different experiments that were run using nanopore. One of them was actually doing the sequencing in the clean room. So we actually had a team from Cornell come out. I'm forgetting his name, Chris Mason. Yeah, him and sort of his team came out and were able to show that we could do sort of live sequencing from samples that were taken right then and there. But other than that, we're not using nanopore too much.
Thank you.
Yep. All right. So moving on to the next project, maybe a little bit more relevant to the sequencing realm. So as Lisa mentioned, first day, sort of we've been collecting samples from missions since the Viking missions in the 70s, and yeah, GSAM. And the isolates that resulted from all of those assays have been kept in the sort of microbial archive. So we have over 5,000, actually well over 5,000 now, probably 7,000 species or strains that we've collected throughout the lifetime of all of these missions. A lot of them have not been identified. And sort of the technique that we were using originally was 16S RNA sequencing. And so we put together this proposal to go through and identify the unknown organisms using a hybrid sort of 16S and MALDI-TOF based technique, which is a sort of time of flight mass spec instrument that's used for identification, and then compile all of that data and sub-select some interesting organisms that we want to do whole genome sequencing on. And sort of the motivation for doing whole genome sequencing is to get better species identification, doing functional characterization, subspecies profiling, and then sort of most importantly, develop a reference database that's representative of the strains that we're picking up when we do metagenomics on cleanroom environments, and so establish that whole genome database that can be used to then map metagenomics data sets onto. As Joel mentioned, the project was called GSAM. It took a lot of nifty thinking to come up with acronym. NASA imposes doing that. But it stands for Genome Encyclopedia, Spacecraft Associated Microbes. And I'm just showing all the different missions that we picked up isolates from, starting with Viking, all the way through to Mars 2020. So we did not work with InSight and Mars 2020 isolates for the scope of this project, mostly because there was just too many organisms, especially for 2020. So this is sort of the high-level overview of the workflow that we followed. So we would revive isolates. These were kept in frozen stocks at minus 80 degrees. They were either in glycerol or in cryobead form. We would initially do identification using MALDI-TOF. So that was sort of the first technique. And if we didn't get good identification with MALDI, we would do DNA extraction and then send it out for Sanger sequencing. And then once we collected all of that data set, we would sub-select for recurring organisms that we were seeing across multiple missions. potentially novel organisms, and then abundant isolates to do whole genome sequencing on. And then once we collected the whole genome sequencing data, the idea was to do some functional genome analysis and then pan-genome analysis as well. So what is MALDI-TOF? Like I was mentioning, it's a mass spec instrument that's used for bacterial identification. It's pretty ubiquitous in sort of clinical labs. And we got this instrument 5 years back now. And initially, as you can imagine, when we were running these isolates, we were not getting good identification because the reference database that was used was sort of very clinical isolate biased. And so we had to establish an in-house database that's representative of some of our organisms to be able to get sort of better identification. So the methodology that was used, we did formic acid tube extraction. There's 2 different techniques with MALDI-TOF. You could do a sort of direct inoculation of the cells onto a metal target or go through sort of this protein extraction. And so we would treat it with ethanol, vortex that, decant, and then add some acetonitrile and then formic acid to sort of lyse open the cells. And that all gets put onto a metal target, which is then overlaid with matrix, and that's what gets fed into the instrument. Some of these slides is actually provided by Akemi, who's online now, so thank you. She also did a lot of work with the 2020 isolates and some of the identifications, MALDI-TOF identifications for those organisms. And she's much better at building charts that are visually appealing than I am. So the isolates were analyzed by mass spec in replicates of 4. The spectra are then compared to that reference database. The scores and identifications are examined for sort of reproducibility. And then that's sort of how we establish whether it's a species level identification or genus and sort of our confidence in it. So the scoring strategy, I believe there's, okay. So the criteria that we were following for using this instrument and sort of ranking the scoring was to have consistent identification in 3 out of the 4 replicates that we were running. And so you can see that in this example over here. We're getting actually all 4 replicates identifying to the same species and strain, which is surprising. And then the scores are their sort of log confidence scores that are basically comparing the spectra to each other. And so above a 2.2 means that it's a confident species level identification, and then above 2.0 is a genus level ID. So that's sort of the criteria that we followed. And these are all the sort of different bins that we were using to categorize all the different possibilities of results. So this slide is just talking about the database itself. So the Bruker reference database that was provided by the manufacturer had about 9,000 strains, most of which were irrelevant to us. And then the in-house curated database that we developed had about 509 bacterial strains. And sort of at that point, at this point, we were getting about 85 to 90% identification with some of the unknowns that we were running. But again, that percentage is very dependent on the mission. As you can imagine, there are different clean rooms used, different personnel, and so the microbiome totally changes. The way we built the database was using isolates from multiple different missions. But again, it's sort of an ongoing effort to continuously add new species that we're picking up. How am I doing on time? Okay. So 16S RNA Sanger sequencing, I'm sure you are all familiar with this technique. So we were using 3 different primers. We were originally using 2. We bumped it up to 3 just to get better coverage in the overlap region. And as robust and sort of gold standard as this technique is, it's very limited in species identification, specifically within the Bacillus genera, because there are very closely related Bacillus species that share sort of 99% sequence similarity in their 16S sequences. And so that's why we wanted to switch to using MALDI-TOF, which has much better resolution. And then, of course, whole genome sequencing can even give you sort of better confidence in species identification. So this is high-level sort of genus-level diversity of about 1,200 isolates from a variety of different missions that we used in this project. As you can see, a majority are Bacillus, as expected. We have some potentially novel genera here. And then, surprisingly, there are some non-spore-forming organisms. This has been of interest to us for some time, and looking to do some more studies on that. It's surprising because the process that we use to isolate these organisms goes through a sort of heat shock step, which you would typically think vegetative cells can't survive, but we're picking up a lot of staph using that. And then this is the species level diversity within the Bacillus genus. So lots of potentially novel Bacillus. A great majority is Megatheriums, something that comes up across multiple different missions. And some novels that we've actually described in-house. So Hernecki is one that was described by our group. And there's got to be a couple others. Okay, moving on to the whole genome sequencing part. So we did some mostly automated DNA extraction using the Maxwell instrument. If Maxwell didn't yield good, sort of didn't give us high quality DNA, we went through a manual. And then we obviously did a quality check as well for concentration, as well as having high molecular weight DNA that can then go into library prep. So once we prepared these samples in-house, sent them out to a sequencing vendor in the UK, and they gave us fantastic data back. We used a paired end platform, so we did 250 base pair reads. We got about 30 to 50x coverage, which was good for doing assemblies for most of these. These are sort of the parameters that we used for quality length and filtering. And then the criteria we had for genome assembly was to have less than 100 contigs, ideally. And then L50 was used to sort of assess the overall quality as well. And this is just a diagram showing the sort of process for whole genome sequencing. So we're fragmenting a bunch of DNA, finding overlaps to do the assemblies, and then so the second step is working with individual reads, and then in the third step, you're combining these concatenated reads into sort of larger contigs, and then in the fourth step, you're doing that again into larger scaffolds. And so when you're scaffolding, you're filling in the gaps in between the contigs, and sort of estimating how large that gap is, because we basically have no sequencing data to cover that region. Once we got the assembled genomes, we had to do some identification. And so we used the GTTB database. This is out of the University of Queensland, Australia. And they have, I think this is an old picture, but they have about 62,000 species of bacteria represented. And as you can see, 317,000 genomes. And the real power of this sort of database is a lot of these genomes are not from culture-based organisms, but from assemblies done using metagenomics or single-cell sequencing. And so it sort of has more breadth than some of the other culture-based databases. So GTBTK stands for Genome Taxonomy Database Toolkit. Consists of 3 steps. So it goes through identification, alignment, and then classification. Obviously a more robust technique than 16S sequencing because you're working with a much larger sequence. And it also has information about functional genes, which can assist in sort of doing some of our annotation work. So this is the identification of the 200 whole genome sequenced isolates that we sent out. And this is categorized by mission. This slide was also put together by my wonderful intern, Michelle. And as you can see, a lot of it, Bacillus is sort of popping up across all of the different missions. And there's sort of a lot more diversity within MER compared to some of the other organisms, but following sort of the same what you would expect based on sort of the 16S identifications that we had. And then these are showing the sort of species across different missions within the Bacillus genera. Again, Megatherium is a common one that's coming up. And yeah. Sort of next steps for this project. So we want to perform some more genomic comparisons at the strain level to identify patterns between species across multiple different missions, look at novel organisms, looking into unique genes that may sort of give us a hint into survivability, and then apply sort of the metagenomic read mapping against some of these genomes to see if we get better mapping compared to some of the other publicly available databases. And that's all I have.
That's great, Armin. I know that there's been some conversations in the chat, but everyone feel free to unmute your mics and ask Armin any questions that you have.
Hi, Aman. Great talk. This is Wei Zhen. Hi, are you guys still, so you're moving to genome sequencing, whole genome sequencing. Are you guys still use multitop?
Yeah, we're still using MALDI-TOF, and we're using some of the whole genome based IDs to sort of go back to MALDI and correct some of the identifications, because the way we built the MALDI-TOF database was based in 16S, and that's sort of inherently flawed, and especially with some of the closely related organisms. And so we're running into issues now where we have database entries that may be misclassified using 16S, and that's leading to sort of more downstream misclassification.
Okay, so basically you still want to use multitop as your quick analysis for future isolates.
Yeah.
But using the whole genome sequencing to calibrate that.
Yep, exactly.
Okay, thank you.
Yeah.
Brooker's database was nowhere near good enough, right? They had the 8,000 isolates they had from Path Labs, and those are all well-known strains, and obviously half of them weren't well-known. Yeah, this has been a fun project for you. Yeah, this is very cool.
Exactly.
Yeah, and MALDI-TOF too. That Brooker what has he used? A MicroTOF 2 or whatever was it? Or is it a MicroTOF Flex or? Anyway, the resolution on that thing is so garbage anyway. Like that's like current gen. That's like, yeah, you could be using a more high resolution instrument and getting better data off it now.
Yeah,
But
absolutely.
I guess you started the project with it, so you have to finish the project with it.
That's exactly right. The instrument itself is almost obsolete.
Yeah, As far as I'm aware, they stopped selling it, and the service contracts expire, I think, the end of next year for them, so they will be totally depreciated.
Unfortunately.
Yeah.
Thanks all for listening. I think we can hop to Lisa if there aren't any other questions, or I think we're gonna go for a break maybe.
Our break is scheduled at 9:10, so we have 20 minutes. I don't know, Lisa, if you want to get started, and then we'll break at 9:10, and then come back for you to finish up.
Yeah, that's fine. Oh, Armand, I did tell you since I got back from London that in the London subway, on the like escalators, the handrails, each one of them has like a UVC lamp attached at the end. So it's like every time it turns through, it's getting
Oh wow,
blasted with the UV.
really cool.
Yeah. Yeah. Okay.
Yeah, there's a lot of interest on the UV side for sure, and I think COVID exacerbated that
Accelerated it.
so much.
Yeah. Yeah. But I was thinking like the escalator rail, it goes so fast. Like that's not enough time. What's the dose rate? Like how can it be?
Yep. They have phone sterilizers now, UV, all
Yeah.
sorts. The wrong line of work. I should be selling UV sterilizers.
Joel said so many in-house filters now have UV in them. They pay you buckets of money to say their product works.
I like buckets of money.
Yeah. Although I think they claim it works anyways.
Exactly.
Okay, are you guys seeing this?
Yes.
Yeah, we
Yep.
see the PDF.
Okay. Great. Okay. So I'm gonna talk about a project that I've been working on for a few years now. It actually ended last year, which is the genetic inventory of our Mars 2020 spacecraft and associated surfaces. And this task was created. So some of you may have already heard bits of this talk before, but probably not since we like completely finished the task as of the end of last year. And so this genetic inventory, or GI, I'm gonna call it GI task, was created in response to Mars 2020 planetary protection requirements at the project level. And so the project level requirements said that we need to identify, quantify, document, and archive potential pre-launch terrestrial contamination sources. And it's not limited to just spores or any one type of organism in particular. We wanted to capture all forms of microbial biological contamination. And so in order to do this, the method that we chose is DNA-based metagenomics. So essentially, if it has DNA, we should be able to capture it with this method, and it would include bacteria, archaea, eukaryotes, viruses. So we're basically creating a comprehensive microbial placental list is what we called it for Mars 2020. And so the level 1 requirements, also what I have bolded here, is that it specifically called for mechanisms to support round-trip terrestrial contamination. And what that means is that we want to identify contamination that could make it into the cache samples that Mars 2020 is collecting now for Mars sample return to bring back. And so this was kind of the workflow that we implemented. We would collect samples from spacecraft or spacecraft relevant surfaces, process the samples with DNA extraction, sequence, and analyze the sequence. And so the GI sampling approach targeted hardware locations with the highest probability of access to cache samples and areas with a direct vector to the Martian environment. So what that means is that we didn't actually sample the entire spacecraft. We did not have enough resources, and we didn't necessarily need to in order to meet our requirements. And so we were strategic in where we collected samples from. And so the areas with the highest probability of potential contamination to the cache samples would be areas that were kind of external to the spacecraft, such as the exterior of the rover or the second stage, which actually crashes onto Mars. And so also anywhere where there would be a chance for something to become windborne in the Martian environment. And so we categorize the spacecraft into 3 priority levels based on this probability. And what that translated to is that we had high priority areas 1 and 2, and then priority level 3, which I'll go over in the next slide. Here. Priority level 1 was all sample caching system hardware, including the adaptive caching assembly and the core, and any rover exposed exterior surfaces. Priority 2 was the descent stage exposed exterior, also the aeroshell and the heat shield. And priority 3 were everything in orange here. So that includes the crew stage, the exterior, the aeroshell and heat shield, including the parachute, and any internal surfaces that you see here highlighted in orange, so interior to the rover or descent stage. And just something that I did not mention in the previous slide was that a subset of samples were also sent for long-term storage at Johnson Space Center. And so the idea here is that as kind of sequencing and microbial detection technology improves in the future, we may have better understanding of what's in these samples if we were to reanalyze them then. And so we have samples that we have actual swab and wipe samples that are stored at Johnson Space Center. And then we also have DNA samples. Post-extraction, a subset of that is also stored and archived. Oh, sorry. And so this project was kind of a first of its kind because we are working with spacecraft surface samples, and so we dealt with a lot of challenges along the way. Essentially, in each step of our workflow, collecting and characterizing DNA from already clean spacecraft surfaces was not straightforward, and so we've encountered a lot of challenges along the way, but we also kind of gained a lot of lessons learned that could be applied to future missions as well. And so as we break down each section here, the first, or the second now, is following sample collection, sample processing. So when it came to processing the samples in the lab, the greatest challenge, of course, comes from the extremely low biomass nature of the samples. And we knew it would be difficult to extract quantifiable amounts of DNA, particularly from the parts of the spacecraft that went through really strict cleaning regimes, like the sample caching system. And so textbook extraction protocols weren't, I guess, stringent or tough enough for our extremely low biomass samples. And so we added several steps to our extraction process to yield as much DNA as possible. And so the steps include, we used dissociation buffer twice on every sample, and then completely spinning them dry each time. So we would use centrifugation baskets or filters with our swabs and wipes. And then we would use chemical and physical lysis on the resulting elution. And so I think this is what made the biggest difference is that we pooled and concentrated several samples together prior to DNA extraction. And so we would pool them together based on hardware grouping. For example, if we have several samples from the rover wheels, we would just, following dissociation, combine the buffer, concentrate that, and then extract that as one sample, as one DNA sample. And this allowed us to kind of capture hundreds of centimeters squared per sample, and we were able to kind of get enough DNA for sequencing. And of course, the trade-off here is that you kind of lose resolution. But for our purposes, kind of capturing larger areas of spacecraft was okay. It did not kind of mess with our requirements. And the final DNA yield was quality checked with PCR and qubit. We did a manual pre-process and performed an automated extraction with QIAcube, which is a column-based extraction method. It's a little extraction robot that we have. And so lastly, we quality checked with PCR, and if it had detectable amounts of DNA, it continued on to sequencing. If it did not have detectable amounts of DNA or above background, we would pool it with other DNA samples from the same grouping and then quality check it again. And so here is an example of our qPCR results from our rover samples. And so you'll see that for each sample, we essentially had one control. So this project we had so many controls because we were going to see, and we definitely did see kind of, kit contamination or other types of background contamination. And so we wanted to make sure, since our samples were so low in biomass, that we would actually be able to kind of discern between what is likely to be contamination and what is likely to actually be in our sample. And so the line here is our background. This is just from the elution buffer that we ran PCR on over and over and over again. And so you'll see that for a lot of our samples, we got about the same amount of DNA as in the controls. And so for those samples, we did not continue with sequencing, or we would pool them together with another sample and try again. And so this is the rover. We have a few from the descent stage and the cruise stage. And so kind of as we are going through the process of this project, so the GI project actually did not inform cleanliness requirements for Mars 2020. So this is kind of a tech development project. It's required that the project collect information. However, it's not like the NASA standard assay where we'll sample, see what we get, and then go and tell our technicians and engineers like, oh, your hardware is dirty. And so we're not there yet. Rather, this was kind of an exploration of, well, how could we develop an assay that could one day inform actual flight implementation requirements like the NASA standards bore assay. And so, one of the things that we thought is, well, could we set a threshold for our qPCR results? Can we say, above a certain copy number per surface area, that's too much. And so what we saw was that we actually had a large range of control concentrations. And so here, this was kind of our distribution of our qPCR results for negative controls and then the sample. And you'll see that there's a pretty large overlap. And due to a large range of the control, it may not be appropriate to apply an absolute threshold. And so the idea is we could look at relative concentrations. And so look at the concentration that we get from the sample and from the control. And if it's a certain, relative amount above the control, maybe we can say that's unacceptable. But this is all just ideas for future assay development that we learned from collecting actual spacecraft samples. And so from there, for sequencing, we used an external vendor, Fulgent Genetics, who would process our samples. And the requirements were that the sample must contain at least 1 picogram of DNA. So they did some pilot testing for us, and that was the amount that they were able to work with. And they also required batches of at least 40 samples. And so this was the minimum amount that we needed to have a whole sequencing run just for our samples. We didn't want to kind of get mixed with some of their other clients that they had, in case they had higher biomass samples and there would be any carryover contamination. And so our library prep included a linear amplification step that they developed in-house, and we used Illumina HiSeq. And so what we learned from the pilot samples that we sent to Fulgent, that the data that we got back, is that the data interpretation is very, very challenging for such low biomass samples. And so, I mean, the pros of a metagenomics approach is that it is highly sensitive, and we can pick up on DNA, from any organism, but it also, like I mentioned before, it picks up on a lot of noise, from the laboratory reagents that are used in the process. And so with samples as clean as ours, the noise can easily overwhelm the true microbial signatures, and it becomes a more difficult process to find those true signatures. And so what we set out to do was to see if we can find out, what is actually coming from the spacecraft and what is noise due to the process. And so our project quickly became just as much about the controls as it did about our hardware samples. And so we always had our negative handling controls that we would take during sequencing along with our actual spacecraft swabs. We also included negative extraction controls. And so this would span different lots and various laboratory consumables like the extraction kit, buffer, and water. We actually sent samples we would stop at various points in our extraction process and send those samples for sequencing, because we wanted to see, okay, well, was there any particular step that was contributing more than others? And then we had negative sequencing controls, so we asked our vendor to include our negative library prep and sequencing controls. And we also included positive extraction and positive sequencing controls. And so positive extraction are actual cells that we extracted from, and from a mixed community, so that we could see, which groups, perhaps maybe they're tough to lyse, that we're missing. And positive sequencing was just DNA, so microbial community DNA, that we got from Zymo that we sent as part of every single sequencing run. And in the end, we really did just have as many samples as we had controls. I think we actually ended up having more controls than samples, which we needed since it was the first time we had done this. So the last step in our workflow, how am I doing on time?
Have about 3 minutes till the break.
Okay. This gets there's a lot of material here. Should we stop here?
We can take a little bit longer break. So if you guys all want to go ahead and take a 22-minute break and be back here by 9:30 a.m. Pacific time.